thumbs up. Admins, you can unshare screens, and DDA, hang tight, stay on the screen, and we're just about good to go. I check the timing. Hey, we're at 1 o'clock. Okay, here we go. Hello, everyone. Welcome to Loop and Learn T1D speaker series. And it's exciting today. We have guests from Sweden, all the way across the pond. It's Digital Diabetes Analytics. It's a company I've been watching for a while. And then I got real excited when they got a lot of recognition recently. And we have all three co-founders, Lars Cederblad, who's CEO, and also a T1D, Dr. Daniel Espes, or he'll correct me if I've said that wrong, Chief Scientific Officer, and Dr. Per-Ola Carlsson, Chief Clinical Officer. It's quite a crew here, and we're very honored to have you staying up late on Sunday night and joining us. We're a looping community, and so we will do our normal disclaimer that the Loop app is a do-it-yourself closed loop algorithm. And while it may seem obvious, please consult with your healthcare professionals regarding your diabetes management. Remember that this project is highly experimental and not FDA approved for therapy. Therefore, you take full responsibility for building and running the system, and you do so at your own risk. And just gently, if you're looping, nod yourself and say, yep, got it. I'm going to remind you one more time that there's a European group that's called Open Diabetes, and they have a survey that they are trying to fill up with people on DIY Loop or family members of those who are looping. If you have not gone to this site, I'll post it again in the Facebook group, but please help them along. They're just trying to fill their survey. It's significant and will be useful to all of us in the future. And once again, shout out to our amazing and growing website called loopandlearn.org, which started only at the end of November, and it's taking on new life every single day. We also put out a weekly newsletter with updates that you need to know about, information about iOS updates and any kind of hot tips and how do you manage Loop. And check back in, bookmark it. It's definitely worth paying attention to. And if you can, please sign up for the newsletter. It's really very good. It's getting better every single one. So please join us. And so now I'd like to just do the introduction because I'm really quite excited today to introduce you all to Digital Diabetes Analytics. They are based in Stockholm, Sweden, and their catchphrase is turning raw data into knowledge and recommendations through algorithms and machine learning. To me, that's just kind of the essence of what we've been asking for is we have all this data. What do we do with it? How do we make our lives better? They submitted the registration to the Medical Products Agency and are now registered as a medical device operator with a CE marked medical device as of November 2020. That's pretty significant. They were also chosen for the Microsoft for Startups program, which offers counseling and technical expertise. And they received that in January 2021, which is when I started sending them more emails saying, come talk to us. And they've been busy, absolutely busy. Digital Diabetes Analytics is developing a support system that automatically analyzes all the data that we put out on all our devices, uses data generated by continuous glucose monitoring, CGM, generates interpretations of the underlying cause of glucose excursions. Wouldn't that be nice? And provides recommendations on how to optimize treatment. I see this and I get very excited saying, I want this yesterday. Lars Cederblad is CEO and co-founder, and he is an engineer and has been living with type 1 diabetes since the age of 15. And we also have co-founders, Dr. Daniel Espes, Chief Scientific Officer, and Dr. Per-Ola Carlsson, Chief Clinical Officer. And they're both physicians and researchers who have devoted their careers to better understand the pathophysiology of type 1 diabetes and how to best treat the disease. We're in good hands here. Their tagline, as I said, is turning raw data into knowledge and recommendations through algorithms and machine learning. And their vision is to become a leading supplier of digital decision support services and care that enables people with diabetes like us to live life to the fullest and be flexible and improve their glucose control. This is all we've been asking for. When we put out a survey, we asked, what did you want? And we want access to our data. And we want to understand how to use the data we're getting to make our lives better. So I'm going to turn this over now. I'm real excited to listen to this presentation. Welcome, Lars and Daniel and Per-Ola. I'm turning this over to you.
Yes, so thank you very much for the kind introduction and for the invitation. We're super excited to be here and a bit frightened by all the questions that will pop up in the end, but that's basically what we're looking forward to the most. And I don't know, can you see my screen now?
Yes, we can.
Okay, good. So we were thinking we will walk you through what we've been working on over the last few years to give an insight on our view on diabetes and diabetes management and perhaps most of all data management and what you can do with all the data if you use them wisely. And as mentioned, what we sort of set out to do is to turn all of this raw data into knowledge and to make some use of all the data instead of just sampling data over time. And let's see if this works. So we're sort of a trio with somewhat different experiences. As mentioned, Lars lives with diabetes, and me and Per-Ola has devoted most of our lives to type 1 diabetes and most of all type 1 diabetes research. So we both, although there's sort of an age gap between me and Per-Ola, we both started our careers quite similarly around the age of 22 by serendipity in a lab in Uppsala, studying islets of Langerhans, and that sort of got us hooked on diabetes. And since then, I've been thinking about insulin producing cells or beta cells more or less every day, all the time. So everything I read and everything I read in med school, I was wondering, can this impact beta cells? How would this affect beta cells? Can it have an effect on beta cell destruction, beta cell regeneration? How would this affect diabetes and so on? So that's how we've been tossing and turning around with beta cells. And we've conducted studies, both experimental and clinical studies on beta cell regeneration, beta cell rescue therapies, imagings of beta cells and stuff like that. And in parallel, we're working as physicians. So in that end, we're more or less battling completely different things, not so much about beta cells. There's no talk about rescuing beta cells in the clinic. There's no talk about regenerating beta cells, but there's more talk of how can we connect this sensor to our system? How can we get the data? How much insulin should you give? Do you need to prescribe any other medications and so on? So there's a bit of a gap between the ongoing research of type 1 diabetes and the current practice. And unfortunately, there's not much attempt to actually cure or better diagnose diabetes at onset, at least not in Sweden today, but hopefully within the coming years, there will be a change in the scenery also for the clinical treatment of diabetes. But from that point of view, we've been telling ourselves and we've been telling the patient community that within a few years or within 5 years, there will be this new and really amazing therapy, or there will be stem cells or insulin producing cells derived from stem cells. But we've been using the same term 5 to 10 years for the last 5 to 10 years. So then when we met Lars, we realized that there's actually a whole lot that we could do right away without waiting for all these new developments, but just to improve what we already have at our hands today. So perhaps Lars, you would like to give a short introduction about your perspective on diabetes care. And
Yes, thank you, Daniel. So actually, as was said, as Johan said in the introduction, I've been having type 1 since I were 15 years old. I have an engineering training. I was actually in research in systems engineering when I was at the technical university a long time ago. Then I went into various industry positions, and for the last 10 to 12 years, I've been running my management consulting company in software development and agile development, and at the same time living with type 1. And when I started using the CGM, the Libre system actually first, I started wondering, what can I do with all this good data? Because there are so many things I could see in the data that was not seen by my clinician. So I started going back to programming myself. I was applying both algorithms and some machine learning practices and found some amazing results. And then I actually, by coincidence, met Per-Ola at another meeting, and we started chatting. And Per-Ola, you invited me to Uppsala University, and there was Daniel, and then we decided to start. But really going back to the core of the company, what can we learn out of the CGM data that is not taking into account today? And Per-Ola, you said that you have better decision support in your car, which is not the newest one, I think. Better decision support in your car than you can offer to your patients at the university hospital in Sweden, which is actually something that I brought with me through the development. And also I think I've been rather lucky in my diabetes career. I'm not on pump. I still use the multiple daily injections. I managed my diabetes throughout these years quite okay. But I know that there are maybe 2/3 of the Swedish type 1s that doesn't reach the treatment target that is set up by the government. So that is really what makes this important for me, really, to contribute to change the life of people and improve the metabolic control. And I was so happy when we formed this company together, the three of us. Daniel, I bring the word back to you.
Yes, thank you. So I think we sort of formed the company out of frustration, both from a patient perspective and also from a clinician perspective, that we have sort of, since the introduction of CGM, been aware that we have all the data, but we don't make any use of it. So that's like our starting point. Like we have to make something out of it, and that's how we set off. And just to give sort of an overview or perspective on diabetes care or diabetes management, as mentioned, we hope and believe that there will be a lot of things developing over the next few years that will change the scenery for type 1 diabetes. We've just divided them into different categories, but of course, if you could prevent the onset of diabetes, that would be amazing, but it doesn't really help people who have already been diagnosed. There's also the case for rescue therapies, that if you at onset, based on autopsy studies, you would have perhaps 30% of your beta cells left, or 40% at onset. But if you could rescue those, then you would be in a sort of prolonged honeymoon phase, which would be great. Could perhaps be done by mesenchymal stem cells or different types of immune modulation or immune suppressive therapy. And then there's also, of course, the hope for regenerative therapies, which would be applicable to basically all patients with type 1 diabetes, or everyone living with type 1 diabetes. If you could either proliferate beta cells or make cells de-differentiate into beta cells, or if you could replace the lost beta cells, which is actually possible today. You can either transplant the whole pancreas, or if you want a slightly different approach, you can transplant just the islets of Langerhans. And of course, for that purpose, you could also use insulin-producing cells derived from stem cells, which is currently not in clinical practice, but perhaps within a few years. You could also use encapsulation techniques in order to transplant beta cells. But then, of course, you also need to manage diabetes today, and that's sort of where we're at right now. And that's what we like to call, you can refine the current therapy, which is insulin therapy. There's, of course, been a development of new insulin analogs. And since your community is all set on pump therapy, and perhaps do not, there's not a lot of need for new analogs or long-acting insulin analogs if you have an insulin pump. But if you're on an MDI, that's quite important to have refined insulin analogs. And another quite intriguing topic would be if you could have a disruptive insulin, which could basically sort of act as a biological insulin pump, that you could inject insulin that would be sensitive to the glucose concentrations in the subcutaneous tissue and release insulin accordingly. Then, of course, there's the development of the CGM technology. And within the coming years, there will probably be new sensors measuring other aspects. Perhaps ketones is one of the things Albert is working on. There could be also sensors to detect other hormones such as cortisol or stress levels, which could be used to refine different systems. And of course, as you're well aware of, also closed loop pumps, which is also becoming commercially available, at least hybrid closed loop pumps. And then, as we have set out to work on, is to sort of automate the data analysis in order to make better use of all the data and to improve diabetes care, which is what we will focus on from now on. So regarding the frustration and the use of technology, despite that in Sweden we have quite well developed diabetes care with certain diabetes teams and diabetes clinics. And we also have a quite different healthcare system when it comes to funding as opposed to the US. But in Sweden, insulin is free of charge. And if you want to use a CGM system, that is for most patients with type 1 diabetes also free of charge. The use of insulin pumps is sort of also free of charge. So there's been a dramatic rise in the use of CGM technology within the last few years. So for patients with type 1 diabetes in Sweden, more than 80% would use a kind of sensor, either CGM or FGM. But as you can see also, during the same time period, this is the amount of patients that reach the set out goal for HbA1c, which in Sweden is 52 millimoles per mole, which would be similar to 7%. So despite the extensive use of technology, we have only had a slight increase in the number of patients that actually reach that target glycemic control. So we are not really making use of all this technique, although it's of great use for the individual, but we can sort of do a lot more with this data. And one question you often ask is, so how hard can it actually be? If you have all the data, why can you not make proper decisions? But since most of you or many of you who are listening live with diabetes, we don't need to explain to you, but this is something we often get asked about from politicians or other people that we're presenting to. They would ask us, but if you have all the data, why would you need anyone to analyze it? But as you all know, everything you do would impact your glucose control, everything you eat, everything you touch, basically. If you sleep, if you don't sleep, if you are physically active, and so on, and infections and COVID, or the stress of COVID, or the sickness of sitting at home due to COVID would also impact your glucose control. And from a clinical point of view, we would basically just use the ambulatory glucose profile. So instead of sort of analyzing all the data and all the things that happened over the last few months or the last year, depending on how often you see your nurse or physician, then we would sort of focus on these mean values and time in range, which of course are good metrics in order to summarize glucose levels, but it doesn't really give any insight on why glucose fluctuates or what you should do to avoid that. So that's sort of what we set out to do. So we want to use all these data that you can collect from sensors and to analyze it automatically and to generate some type of clinical knowledge and to identify root causes for glucose excursions, to give weekly summaries, to do individual glucose profiling, and also to present sort of a trajectory towards improved glucose control, and to find different risk scores and to find areas of improvement. So we've developed a lot of different setups or algorithms, if you will. Some of them are based on machine learnings and some of them are pure algorithm calculations. And we will go through, or we will present you the system as it looks up currently. And these are some of the analysis that we have developed so far. So we've focused a lot of our energy on the things that we can actually alter, such as the insulin basal pressure. We can identify the risk of hypoglycemia or the risk of hyperglycemia, the impact of meals and the impact of hypoglycemia. We can identify what happens after hypoglycemia, and we can identify root causes of both hypo and hyperglycemia and how you recover from it. So in order to give an insight on how we have developed the system and how we have validated the system, we will focus a bit more on hypoglycemia. This is a quite distinct event and is common for most of those who live with type 1 diabetes, and is often a fear for those who live with someone with diabetes, and is also a great fear for physicians treating type 1 diabetes. So this is also a sort of a given thing for you. So we would define hypoglycemia in Sweden as a blood glucose level below 3.5, which seems quite dramatic since you use another units for hypoglycemia, but that would resemble 63 from your point of view. And this is a sort of a graphic presentation of what happens when you have hypoglycemia. It's the words are in Swedish, but I think you can understand them anyhow. You can get tired and you get mood swings or headaches, you get pale, sweating, and so on. And even though hypoglycemia is something many would experience on a daily basis, hypoglycemia is also actually something that can be a cause of death in type 1 diabetes. And also the fear of hypoglycemia or actual hypoglycemia can also be an obstacle for achieving optimal glucose control. And I think that from a clinician's point of view, we often sort of, we would look at the time below range, or we would try to identify hypoglycemic events, or perhaps more often just bluntly ask, do you have hypoglycemia and how often? When can you sense that the glucose levels are low? Do you experience unawareness and so on? But we have quite few tools to actually handle hypoglycemia, to identify why does hypoglycemia occur. So what we wanted to do was to sort of characterize the underlying cause for hypoglycemic events. And again, the scale is not really US friendly. So this is the orange line would represent around 63 milligram per deciliter. But it's actually the shape of the curve that's sort of the take-home message here. So based on a lot of hypoglycemic events, we try to characterize these into different underlying causes. And given that we started out with just the CGM data, with no other input data, no patient history, no registered insulin doses, no registered carbohydrates and so on, no information on physical activity. So we tried to sort of decide what can we read out from just the glucose data. And then we decided that there's sort of 3 different categories or 3 underlying causes that could be determined by the type of therapy or the insulin therapy, which would be resembled by high basal insulin pressure, which is often a sort of a slow drop towards a hypoglycemic event, could occur during night, perhaps more often during the night than the day, but can basically occur at any given time point. It could be due to an bolus at a meal. or it could be overcompensation of hyperglycemia with a quite fast drop of glycemia, which is then sort of bounces back up due to perhaps overeating, or in this case, most likely not a peak oil, but overeating. So these were the sort of three main categories. And then there could be other causes, so there could be sort of additional things that would impact glycemic control, but these were the main categories that we tried to categorize. So we did sort of manual interpretations of more than 5,000 hypoglycemic events from patients with different types of therapy. Could be MDI, could be pump. It was both adult and pediatric patients. Then we used for those who actually did registered insulin doses and carbohydrate intake, we used that as sort of a validation set in order to get as close as possible to the sort of the actual event or the truth of what actually happened. And based on these manual interpretations, we started to train and test different machine learning models. First of all, we sort of screened different available models in order to identify those that had the best performance for that type of data. And the model was only aware of the actual glucose levels and the time of day. So we tested these different sort of out-of-the-box models and found that they were actually worked quite well. But then we also started to build a customized model based on a convolutional neural network that actually worked the best for classifying these events when looking at all the different categories. And for an average value, it was quite close to the already built model that was called inception time, which worked well, but was not as good at identifying boluses at meals. So we sort of tried to further enhance this model. We looked at the importance of data, or actually more or less the amount of data. You could see that when only feeding the model with a small number of classified hypoglycemic events, the outcome is sort of by random, but when increasing the amount of data, also the stability of the model increases. And quite interestingly, if we would add more data, tabular features could be like, do you use MDI or use insulin pump? What type of insulin are you using? Age, disease duration, stuff like that. Doesn't really impact the outcome that much. So the model actually sort of only needs the glucose levels and the time of day. Or actually, time of day is not that important either. When we did the manual classifications, we thought that the time of day was really important because we sort of read into the data, oh, this is perhaps a lunch, or this is during the nighttime, and you sort of paint the picture of what could have happened. But for the model, that doesn't really matter. They would just look at the glucose data, and that would give sort of a similar performance score. So when we had trained the model, we then did an overall test, or a test performance with new hypoglycemic events, but from sort of known patients that had been in the original cohort. And we got an overall test performance for classifying the underlying root cause of somewhere about 90%. Then we repeated the same thing with hypoglycemic events, but from patients that had not been in the original cohort, meaning that the model or the algorithm had not seen any data from those patients. So even though it could theoretically learn to identify singular patterns for patients, this was based on the new cohort, and we achieved sort of a similar overall test performance score of somewhere about 90%. So this is actually quite impressive for a model, given that there are so many different things that could impact the glucose curve or the glucose levels. Right now we're actually trying to do a sort of a web-based trial, because for the manual classifications of hypoglycemic events, it was initially me and Per-Ola who did the classifications, and then we used an external expert board of 5 clinicians, both pediatricians and diabetologists. And then we used the validation data from patients who had actually registered insulin doses and carbohydrates. But now we're also running a web-based trial in order to try to identify how well can clinicians actually identify root causes of hypoglycemia. And similarly, we're planning now for a trial, web-based trial also for people living with type 1 diabetes in order to try to classify hypoglycemic events in order to identify underlying root causes. But we do not have the data from that yet. But this is something that we think is quite interesting, because for diabetologists or physicians working with diabetes, there's not really that much instructions of how to interpret CGM data. We have been very focused on identifying mean values or putting data together into time in range and stuff like that, but not that much training on how to actually identify patterns in the glucose data, as opposed to for cardiology and stuff like that, where we would have strict rules for that. So we're quite eager to see how clinicians actually interpret this type of data and how that would match the interpretations from people living with type 1 diabetes. And so from this type of training, we've developed then the different algorithms for hypoglycemia. We've also done similar work for identifying hyperglycemia and recovery after hypoglycemia and so on. So by using this data, we're now developing 2 different systems that will be communicating with one another. One is directed towards the healthcare professionals, physicians and diabetes nurses, in order to automate analysis of data. One is intended to be directed to individuals living with type 1 diabetes, or parents, or a close spouse or relative to one living with type 1 diabetes, in order to analyze the same data and to give the same type of insights, but to be presented in a sort of a to present underlying root causes and what to do differently, and also to identify areas of improvement. And we will get back to that later. So I guess there will be questions in the end, so we will just continue to ramble on, and we will give you an insight on the clinical overview, or the system intended to be used by clinics or physicians. So we've built the system so you can use it to view all patients at once or individual patient reports. And this is the clinical overview, which is intended to use as a sort of a optimization tool in order to identify either patients in need, or patients that need a visit, or patients that are doing great on their own and do not need a visit and do not want to be disturbed. So we've used Power BI now to visualize all of these algorithms, which is one of the collaborations with Microsoft. And again, this is the estimated HbA1c, and it's not US friendly, but it's at least color coded. So green values mean you reach the target, red values means you're above the target for HbA1c. In this view, you can also see for a clinic how many patients are there at a given date and time, and you can use the time slicer to look at different periods of time. But what you actually get is sort of, as a clinic or community for that case, how you're developing over time. Is the number of people reaching target glycemia increasing or decreasing? And of course, you can also identify the list of patients and identify those with the lowest or highest HbA1c, and you can use that either as a prioritization tool or whatever you want to use it for. You can do the same for the timing range, which perhaps would give a better insight than the HbA1c. So you can identify patients with a high timing range or low time below range, and you can compare different time periods. For instance, during a pandemic, are things getting better or worse? You can also identify from an individual patient perspective, the basal, what we call the basal pressure estimate, which would be, regardless if you're using a pump or MDI, this is sort of a estimate of what happens when you're fasting. So we've tried to derive as much fasting time out of the day as possible. And the nighttime is, of course, for most people, the longest stretch of time where you would be fasting. So you can then sort of analyze what happens with glucose levels during fasting, which we would then call like sort of your basal pressure. So this scale ranges from in positive levels, which is not to be confused with positive insulin on board, which is probably what you would use in order to identify or to analyze your basal pressure. But in this scale, this is sort of resembling the level of glucose and level of glucose changes over time. So 1.2 means that glucose increases by 1.2 millimoles every hour, which is of course not favorable if you want to achieve a good glycemic control. On the other spectra, this means that whenever you're fasting, glucose decreases by 0.8 every hour. And from this clinic overview, you can then identify patients at each end of the spectra and use it as sort of in order to prioritize. And since this is not a favorable sin, and this is either not a favorable sin, you can sort of identify those patients and make a clinical decision on whether to change therapy and so on. Can also just mention that from screening a lot of data, we have sort of identified that a common denominator for those with a optimal glucose control is that you often have a slightly negative value, meaning that during fasting, your glucose levels would slightly decrease. This is something we're now trying to clinically validate, but this is just what we've found so far. With the same analysis, you can find or identify patients that have a high postprandial delta, meaning that after a meal, you would have higher glucose levels 2 hours after the meal as compared to before. And this might sound quite simple, but the sort of beauty of the analysis is that we have constructed a filter that can identify meals without you registering carbohydrates. So based on the CGM data, we can identify meals, and that's sort of the data that is presented here from a clinical overview. So for this clinic, what you actually strive to identify is you have a lot of patients with a high glucose delta between meals, meaning that you need to sort of alter your bolus at a meal strategy. And we can do the same for hyperglycemic event based on the number of hyperglycemic events from this given time period and the most likely root causes of that. And similarly, also as we described for hypoglycemia, you can identify the patients, and if this was for a real clinic, for each bar there would be an identifier for that patient here, which I probably should have mentioned for the first slide. But each of these bars would represent one individual. So this is a time period of, I'm guessing, 9 months or something like that, and we can identify more than 200 hypoglycemic events. And for this individual, the majority of those were due to high basal pressure of insulin, which can actually be quite easily altered if you're aware of it. And based on all of the analysis that we run, we can also present all of the data combined in what we call a glucose severity category, which takes into account both the actual glucose levels and also the rate of change of glucose levels. So instead of looking at either time in range or HbA1c, we've constructed this new category or classifier for glycemic control. And it runs on 2 different scales. So you have the glucose distribution profile, meaning your actual glucose levels, and the rate of change profile. If you do not have diabetes, you would end up in this category, which is 1, 1. And we're well aware that we need assistance from a graphic designer in order to make this perhaps more enjoyable, but this is where you would end up if glucose control were sort of perfect. If glucose control is sort of off the charts, it would be in the upper right corner. So from this sort of chessboard, you can then, from a clinic overview perspective, identify the number of patients for each category. And if we were running our clinic of our own, which we are sort of starting with, we would of course use this as a tool in order to identify patients in need of care or need of assistance. So ideally there should be no red dots in this chessboard. Yeah, and continuing to ramble, we will now step into the individual patient report, which will give sort of a similar view, but I was thinking we can then look perhaps more into detail to some of that, and then we will skip some of it in order to get to the Q&A. So this is, if you would look at an extended time period for one patient, this is 3 months. This is sort of the raw data. This is all the CGM readings. Doesn't make any sense. Sort of looks like a seismograph, but this is what we're currently working with, and then you can derive it into time and range and stuff like that. So this is something we're sort of trying to get away from, because this doesn't really make any sense. From a day-by-day view, of course, you need to review the CGM data, but when looking at a larger time span, that doesn't really make any use of it. So in the summary report, of course, you would have the average glucose levels. You would have the estimated HbA1c level. You will have the variability or CV values and standard deviation. You would have the time and range, but then you will have the algorithms or the analysis that we're sort of focusing on, which would be the basal pressure estimate, which would be derived from average levels from the last 3 days, but it would be presented as a one-day value. So you can see how the basal pressure would vary from day by day. You would see the number of hypoglycemic events, in this case, 81 hypoglycemic events over 3 months. And basically all of them were due to a high basal insulin pressure, which is also sort of presented from the basal pressure estimate. You can look at the time in range for that time period. You would get the average time in range for that period. But that's sort of old news. And then looking at the basal pressure estimate, as mentioned, this doesn't take into account if there's a pump or an MDI treatment. And if you have an insulin pump, of course, you would have different settings during different time periods of the day, which needs to be taken into account, which needs to be sort of customized for that patient. But if you have used MDI, that's not really available for you currently to have some sort of estimate of your basal pressure. Prior to the use of CGM, you would say that, okay, the first value in the morning, that sort of resembles your long-acting insulin dose. If you wake up with high glucose levels, then you need to increase your long-acting insulin, which you, of course, would know that it could be high levels in the morning can also be due to hypoglycemia during the night and recoils or the dawn phenomena and stuff like that. So we used to live with this sort of simplified view for MDI treatment, but when analyzing CGM data, we can also, regardless if you use MDI or pump, try to find the right basal pressure estimate. And as mentioned, ideally this should be like in a perfect world, when you're fasting, glucose levels would just stay the same. Then the basal pressure estimate would be 0. But we found that that's not really the case for at least most patients that actually reach target levels or near normal HbA1c levels. They often would have a slightly decreasing glucose levels during fasting. But that's something you can, of course, optimize from an individual patient perspective. And we've also constructed a filter that can identify meals based on only the CGM data, regardless if you enter your carbs or not, or if you use an app in order to take snaps of your meals, or we can still try to identify them. And what we can find for this time period of 3 months we can identify in total there were 135 meals. There were 43 breakfasts that could be identified, which is, of course, there would be expected that there should be 90 breakfasts if you have breakfast every day. So if you had an optimal dosing of insulin to that meal, that would not really leave a mark on your CGM data. So that would turn up as no meal, meaning actually that's sort of an optimal insulin dosing for that meal. And that's not something you need to change, because that was perfect. But then for the meals that we actually can identify that could be optimized, then we would present the preprandial and postprandial meal, glucose value, the delta value for that meal, and the peak value for those meals. So then you can sort of break into details. Okay, if you have breakfast that we do identify, are those on weekdays or work days, and then you can review the strategy you use for your meal dosing or insulin set meal. And we can do the same in order to identify hyperglycemia and try to figure out what's the underlying cause for hyperglycemia, which could be, in this case, there was no lack of basal, which is probably due to the quite high basal pressure, which would more or less lead to hypoglycemia. But from that perspective, from insulin therapy, you can either have hyperglycemia due to the lack of basal, and in that category, of course, there's a lot of contributing factors such as stress, there could be infections, there could be like any given thing, but most of the time you would sort of correct that with increasing basal. Or there could be due to hyperglycemia after meals, which would be the lack of timing or lack of amount of insulin at meals. Or there could be due to that you are over-ingesting carbohydrates after a hypoglycemic event, or that you have a hormonal recall after hypoglycemia. So you can sort of identify what's the driving cause of hyperglycemia. And in this case, there were not that many hyperglycemic events at all. And then, as mentioned, for hypoglycemia, we would do the same thing. We would classify underlying causes. You can split them up by the time of day. You can see the total distribution of hypoglycemia. And in this case, the dominating cause is the basal pressure. And then sort of a background for the glucose severity score is actually based on histograms of glucose. And this is sort of the percentage of time you would spend at different glucose levels, and also the proportion of the total time for the rate of change in glucose. And from that histogram data, we can sort of categorize all of the weeks into one of these categories. And despite hypoglycemic events, this is actually quite impressive glucose control for this patient. So this is sort of a case study for another patient. And from our traditional clinical view, we would sort of review the ambulatory glucose profile. We would look at the time in range. And this would resemble an estimated HbA1c, which would be 7%, which would actually be sort of like from a physician point of view, we would say, okay, you've reached the target, so things are moving well. There's no need to change anything. But actually, this patient, looking from a glucose severity category point of view, would be a red flag saying that something is not really working well here. So then looking at the different types of analysis that we can do with the data, we would find that, first of all, the basal pressure is quite highly variable. And most of the time during fasting, glucose levels actually increase quite a lot. We can see that also the postprandial glucose levels after meals are not optimal. And the peak values after meals are quite high, especially during dinner. And despite that, there's also quite a lot of hypoglycemia. And most of those are due to overcompensation, meaning that due to the lack of basal insulin pressure, you need to correct high glucose levels with short-acting insulin, which would lead to a highly variable glucose. So regardless of the HbA1c level, which does not take into account any fluctuations of glucose, you can sort of have quite different glucose profiles, which we can then identify by this glucose severity category, and by the different types of analysis, you can sort of identify what are the driving causes of that and what to alter. And so starting to get tired of my listening to myself, we'll get to the end by this. So we're sort of a cross-disciplinary team. We are the 3 founders. We, of course, have a lot of people who now know a lot about machine learning and IT, and we have a great support from Microsoft in our endeavors. That's why we sort of use Power BI in order to visualize all the analysis that we now are developing. And feel free to contact us if you have questions that will not be answered in the Q&A following this, or visit our website or LinkedIn. And by that, I think we will end that presentation. So we will leave room for everyone to discuss.
Joanne, you're muted.
Okay, wow, very interesting. I'm going to ask all admins if they'd like, they can turn their videos back on and their mics if they want to do some questions. And Cass, if you would jump in and start bringing, we've got a lot of questions. I'm scrolling back up through the chat from both YouTube and the Zoom. So Cass, if you want to jump in, I'll let you start.
Sure, I'll lead right off with Rachel, our love, Rachel Gerlin. She asked, we'll make her statement first and then her question. She said, some of the data seems like it would be difficult to digest and make actionable on behalf of the patient. How are you guys making data consumable for the everyday person as outside of the clinician?
Yeah, so the stuff we showed right now is only the views based for the clinics, and we're currently developing a mobile app that is more suitable for everyday life and making quick decisions or making readily digested data. And we will have that labeled within the European Union as a CE mark product during May. So we have a lot of that already on the way, but we decided not to show anything of that right now because we're sort of in that process in order to develop it.
Mm-hmm. And does that give the patient access to run analysis on their data on a daily basis, a weekly basis, monthly?
Yeah, so you can actually, when starting it, you can feed it with as much data as you like retrospective data. And you can have the analysis set for a week, a day, or a year, or whatever time period you would like. So then you can also alter your target levels, and it will sort of find the areas of improvement that you need to work on in order to reach your target levels.
Got it.
So say if you have high variability, that would be where to start, as opposed to trying to just reduce mean values.
Mm-hmm.
It will be more or less the same analysis as in the clinical report, but it will be digested so that we highlight what parts needs improvement, so that you can get specific advice on that part. And then for the next analysis you are doing, you can then compare to the former analysis to see if you have improved and if that was based on following the advice or not.
Is the reason you're using CGM data, it seems like you're using that only, is that because you don't have access to pump data, or a lot of people are using injections instead? And are you seeing this in the future being more people going on to closed loop systems, so they'll be on pumps that are automated?
So
yes,
I can start. So we started out with just the CGM data, because that is sort of the baseline. If like every patient would have access to CGM data. Then like next level would be if you have MDI, that you actually do register your insulin doses or carbohydrate intake, or hopefully now there will be a widespread use of smart pens, then you will get that by NFC. So all of the analysis can actually access or use all of that additional data, but we've sort of constructed the system so it can be run on just the CGM data as well. But of course, the more data you would add and the more type of data, the more features you can derive from it.
And the reports that come out, are they just simply reports that you look at and decide what to do with, or does it automatically generate suggestions that here's where you want to might take a change, or here's what we think your patient should do?
So yes and no. So from a regulatory standpoint, currently it's sort of pointing you into the direction using words that sort of would give you an insight. This is what you should do, but it doesn't really say increase Lantus to 24 units. But basically that's sort of where we're aiming to. But from a regulatory standpoint, that will take some additional work.
But maybe I can fill in on, because I've been following the questions also on the chat in the meantime. We started with a clinic system for the regulatory purposes, exactly as Daniel is describing. And now we're uploading data in batches of data with patient data. But the target is, and when we launch the patient mobile app, we will have real-time data, at least for some CGM brands. But the idea when we go further on is to include more sensors like activity trackers and so on to get even more, not just pump data and pen data, but also activity trackers for pulse and steps and so on and so forth.
Okay, there are a bunch of questions that I think that was what Rachel touched on is food and what you assume is a meal. What about low carb meals that kind of don't get treated and don't show up, or they're treated for later for protein? Your system identifies when you're eating, but how does it do that depending on what you eat?
So the system actually identifies, so either if you log carbohydrates, then like the system would know. It doesn't really take a lot of analysis for that. But so the filter is basically due to, at least from like our perspective, from the data we use or the retrospective data and the like clinical data in Sweden, there are quite few patients who actually do log their carbohydrates. And also, if you do, perhaps you don't do it consequently, or you would log it afterwards or so on. So the timestamp for carbohydrates is sometimes quite uncertain. So that's why we sort of felt the need for this filter. But the filter is derived from the CGM data. So meaning that if you have like a either low carb or no carb meal, or if you have, even if you have like a high carb meal, but you're sort of a pro of delivering insulin, or you're pre-bolusing, or you're doing extended bolusing or whatever, if you don't leave a mark in the CGM curve, we won't identify it as a meal. But that could also be, now we're sort of trying to tweaking that analysis in order to identify those actual optimal meals, and meaning that they were optimal from the point of view that the insulin dose actually took that dent in the curve out. So, but in order to do that, we need to learn more about the user, like the time of day you would normally eat something. So if we can identify at least a number of meals, we can sort of start to learn a pattern that this is where you would normally have your breakfast. And if you assume that you do that every day, you don't skip breakfast, that would be identified as like an optimal meal. But that's sort of where we need the patient app, because that will also open up for an option that you can sort of communicate, or you can have input data. Like you could have a pop-up question saying that, did you skip breakfast today? And if you would answer, no, I didn't, then we would know that, okay, so you handled that quite elegantly.
The prandial analysis also gives the possibility to look at not only if the insulin dose is correct, but also if you have a very high peak that suggests that you eat too much carbohydrates, or that you take your insulin too late, too close to the meal, or even afterwards. So you can get also more information on if you eat well or take the insulin timely to the meal.
So this sounds to me like we had a presentation by Lane Desborough, and he identified quadrants of people with type 1 diabetes, and the upper right corner was the very proactive ones, like the loopers, and that's a tiny percentage. Then you've got 2 quadrants that are somewhere in between, and then you've got a large quadrant that he calls dark matter, and it's those people that are not well managed, don't track their insulin, don't track their foods, don't like to announce meals. Is this the market that you're kind of aiming at, the ones that really need significant improvement?
Yeah, so well, yes and no. So that's the case also in Sweden. So when we analyze like data, we would like one interesting phenomenon. I'm guessing that very few of you use the Libre system, but if you do, then for the Libre you need to actually scan it every other 8 hours, or you would lose data, and you would, and that's a weird thing to start with. Like 8 hours, people need to sleep 8 hours or more. So there's like a given thing. You would lose data, but some would scan it during the night. But then so if you look at, and in Sweden 80% of all users would use the Libre system. So that would allow us to actually analyze quite a lot of behaviors as well, because when you scan it, then you can extract that data from the like continuous scanning every 15 minutes. Now, if you have a newer Libre sensor, it would be more often, but you can identify those that were scanned manually. So from that, you can sort of find a persona for that patient. And what we've learned that there's actually quite many do not scan their sensor, would only scan it once or twice a day. And that sort of gives you an insight on how much time would you spend on your like diabetes management. And we've given that sort of a lot of thought when we are now designing and developing this patient app. Like if you scan your sensor twice or at best 4 times a day, how can we encourage you to use a mobile app in order to improve diabetes care? And that's a tricky part. So even though like from a like one perspective, that's sort of what we're aiming for, to try to get someone who doesn't really care that much about their diabetes to still want to use the system. But that's like that's a tough thing to handle. But one thing that we have thought a lot about is that if you have a high HbA1c, which would be like the metric that's saying like, okay, your glucose control is not good and you're not managing your diabetes, that is probably what you have heard every time you visit your doctor or your nurse, and you know that you don't have to leave a blood sample in one week because you would know. And like coming to that office like for the 5th year in a row or 10th year in a row, that's not much fun. And we did this like just for fun. We did in another study, which was a clinical trial, where we measured if you have endogenous insulin production despite you have had type 1 diabetes for 10 years or more. And then we sort of tried to find, is that linked to better glycemic control, which wasn't. But we also identified what's the best denominator for the current HbA1c level. And that's, of course, the historical HbA1c level. So I'm now taking a detour to say that if your HbA1c is not good and your glycemic control is not good, that's what you will hear at every visit. And what we're trying to build into this app is, instead of telling you that, because you already know, is telling you what can you do that would actually significantly impact your HbA1c that doesn't take 10 hours a day. Like to find the correct basal dose, that would make a huge impact on your glucose control. And if you're on MDI, at least in Sweden, most patients are sort of instructed or told that, okay, you can manage your bolus doses. They could be between 4 or 8 units and stuff like that. But for Lantus, it's 20 units a day. And if you have an infection increased by 2 or 4 units, but you would sort of get stuck on a certain level. And that's, I mean, and it could have passed 5 years and you haven't changed it. But if you would instead increase it to 30 units, or why not 40 units, perhaps your glucose control would be tremendously better. So that's sort of what we're striving for those that, as from that point of view, would be in the dark matter. Don't make optimizations that's sort of pinpointing, but make 1 or 2 changes that actually have great impact on glucose control. But we also, so we're very fascinated by your community, because you have a lot of knowledge and you spend like vast amounts of time on glucose control. And we are sort of hoping that we can also provide something or some insights for your community. But that would be like a completely different point of view. So that's for like the patient app, that's why you can set your individual targets. And depending on your targets, we would look at like this is a multi-dimensional feature, saying like it's not just about time in range. So variability is important, hypoglycemia is important, hyperglycemia is important. And instead of variability, we like to look at it as sort of a stability measure. And then, so yeah, that was a long answer, but didn't really answer your question, I think.
But
I think I could comment a little bit on looking at this from the past. In the past, there was very seldom any individual gave himself extra insulin because they had high blood glucose, because no one really knew that they had high blood glucose. I could have patients that never measured the blood glucose for the whole year, and they had good HbA1c when they came to the clinic, whereas others had terrible HbA1c. And what we aim for is actually to get the patient on the correct basal insulin dose and the correct meal insulin doses. And if so, you will not need so much compensatory insulin doses. That is the main aim, actually, because when the swing starts, I would say, you will get both hypoglycemia and hyperglycemia on and off, and a very high variability.
And actually, Joanne, and back to the black matters again. I think I said in the beginning, in the Swedish scenario, we have about 50,000 type 1s in the Swedish population of 10 million. Out of those, I would say that maybe 15,000 are with an HbA1c below 7%, which is too low. And that's really my target, really, to reach the ones that are above the target level and to help at least a third of them to go to the right side. So I would say you in the lupin community, you are really pros, and it's still a small group, but we aim at helping the big amount of people that are on maybe a simple pump or MDI.
I feel like I have to ask the question because Kenny's not here to ask it. So when you talk about focusing on basal and focusing on meals, right, carb ratios essentially, where does insulin sensitivity play into the GDA mind frame?
So insulin sensitivity is, of course, important, and that's something we sort of derive from the analysis. And like insulin sensitivity would change over the course of the day. So that's something I think I'm thinking that you don't forget that, but I can tell you that a lot of clinicians do forget that, and that's something you need to sort of keep track on in order to make correct decisions. And so that's something we're now trying to sort of draw conclusions from based on CGM data. But for that, you would also need interest of carbohydrates and insulin doses. Otherwise, you can make guesses, but it won't be as accurate as if you have all the data.
Carol asked this very early on in the presentation. Do you guys need data donators?
Yeah, that would be great.
Yeah, absolutely.
I can't
I'd like- I'd like to-
she's snapping right now because she's happy.
I'd kind of suggest possibly, in addition to the approach, and I do understand because we hear from clinicians saying that we don't get what you guys see, which are the people who aren't doing well. And that's true. We're the overachievers, and we're not happy with a time in range that's below 80%, and I would probably say we're not happy with below 90%. So we are high achievers, and we blow the curve every time, and I understand that. But would it also be interesting to gather our data and how we do it, because we're very proactive in terms of dosing, in terms of pre-bolusing, and the data is there. Most of us use Night Scout, but it's all very much real-time data. And I know I would love to participate in the questions are coming in. Do you have any clinical trials planned on glucose control and management? And, you know, we would be at the other end of the people you're working with. I would love to shift your approach and say, we would love a system that knows how we manage it and make some decisions for us because we get tired.
Yeah, so we would love to share data or to view data, and I can say that the data that you collect every day, that could be used sort of like for us from our point of view, like a validation data set, because it would be a as good as it gets data set. And what we would really be interested in is sort of to get you along with like the classifications of hypoglycemia. We have this ongoing debate: who is the best to classify hypoglycemia root causes? Is it clinicians or people living with diabetes? And as a physician, I'm happy to say that I'm certain that it's people that are living with diabetes are the best to actually identify root causes, and especially if you could do it sort of in real time, or near real time, which is the purpose with our app also, is to sort of make a validation set for each individual, meaning like once the hypoglycemia has been resolved, everything is fine, it would be this pop-up question: why do you think you had a hypoglycemic event? And I mean, you would know because it was perhaps just an hour ago. And then we would sort of in real time try to collect that data, and that could be used to further sort of also validate and enhance these machine learning models. And you can use similar approaches for a lot of events that would happen. And that's something, and as mentioned, we're also doing this web-based trial on sort of a fixed set of hypoglycemic events, and it would be really intriguing to see like how would you score or judge these hypoglycemic events, because I'm guessing that you have spent a lot of time reviewing CGM data. So that would be really interesting.
Are we able to jump onto your web-based app?
And not today, hopefully soon. So another thing is,
yes, maybe Daniel, I can jump in.
Yeah.
So we're for the mobile app, we're planning a CE mark in May, and we have our plans is to go for an FDA approval. I hope that I could say later on this year, but I cannot commit to that at this meeting. But sooner or later, absolutely.
I'm a little baffled by, maybe I'm just not understanding from your perspective, why it's so important to identify the sources of hypoglycemia. From my perspective, I just deal with it. And I guess you identify the sources to prevent it from happening, but it doesn't seem to me to be that complicated.
Yeah, so I think that like from that point of view, if you use a like the DYI looping or the like a commercial hybrid closed loop, that would sort of be a issue that's already dealt with, because you would alter basal rate, and hopefully you would not have a lot of hypoglycemia due to high basal rate, or you would make adjustments as you go along. Whereas if you use MDI, that's a very more like the more static life, because once you've injected that long-acting insulin, it's there, you cannot take it back, and you cannot change it until that time has passed. But what often happens is if you don't have a pump that you would log all the data, or you're using MDI, or use like an old-fashioned pump or like regular pump, it's quite difficult to be systematic when it comes to your own life. And if you don't make that adjustment as you go along, then you would repeat like sort of the same mistakes every day. And that's what we often see in the data. So we used actual, like in this retrospective clinical trial, like you would find like a patient profile saying like, okay, you can review 3 days, or you can review data from 1 week, and you would know like, okay, this is your problem. But then when you look at next week, that's the same problem, and the same problem continues. So that's sort of how we decided that we sort of need to identify the root causes and deal with those. So that's actually more or less to enhance our like alterations of basal rate. Also runs on like, okay, do you get a lot of hypoglycemia due to basal rate? And is basal rate like you have a lot of basal insulin, or vice versa? And also like the lack of basal rate can cause hypoglycemia due to overcompensation. So that's sort of what we use the knowledge of root causes for. And it's more valuable for those who do not make adjustments like minute by minute, because then you would have a lot to deal with by the end of the month.
And I need to let you know that those of us who are looping also get hypoglycememia.
I'm quite aware of that.
So, yeah, yeah, we have exercise, or the food hits at a different time, and then we get hyperglycemia. There's some comments here on rage bolusing and rage corrections, because you just go, oh, I'm done, I'm going to do it. And so we're normal in that way. We're just obsessive about how we try to fix it. So
so one of the people who actually work with us at DDA also used DYI. So we've run some like the data, our analysis of that type of data. So we know like how that can be look like. But also like from a clinician point of view, saying that okay, now you have the like you have the Tandem or IQ, and you have the Omnipod will be coming out, and you have the Medtronic systems and so on. But if you thought it was difficult as a clinician to review CGM data before the entry of hybrid closed loop pumps, then you're about to get a lot worse when you have all these pumps that will make all of these alterations automatically without input from clinicians. And then you're sort of supposed to review the data and saying like, okay, did we make the right choice? Is this the right pump for you? Or are you better off now than you were before? And if we upgrade it, or if we choose to switch from Medtronic to Tandem, is this working well for you? So from that point of view, like this is a analysis system. It doesn't matter for the system what type of method you use to deliver insulin. It would just review the data saying is this good or bad. So you can also use it to compare. And like, for instance, if you use a new update or new features saying like, did this improve glucose control or not? And the sort of beauty with automated analysis is that you can look at a lot more features than just saying, for instance, okay, is time in range better or not? Is HbA1c better or not? But you can look at like all of the parameters at once, saying like, is this making improvements or not?
I think it's important to circle back to how the model identifies food that wasn't called out. And like we touched, obviously you spent time on this and touched on it even in Joanne's earlier question. But I feel like a lot of that, can you give us, or whatever you're willing and able to share with us, how the algorithmic model itself identifies a meal?
So it's basically, when constructing that filter, we first of all, we used data from the people who had actually registered carbohydrates and insulin. So that was sort of like the ground truth, saying like, okay, you registered carbohydrates, so that's the ground truth. And that's what we started out with. And then we did a sort of a similar approach as we did hypoglycemia. So like while reviewing that data, we would have manual interpretation saying like, okay, if you log 5 grams of carbohydrate, that's probably not your main meal. It could be if you're on some certain type of diet, but most of the case that would not be like a main meal. So we're trying to identify, and then just looking at that and trying to identify for individual patients, what are your main meals and what do they look like? And then we sort of just ran it through, trying to see, okay, how many meals or events on that glucose curve can we find that looks similar to those meals? And then we'd review those, like again, saying like, okay, for the events we did find, were there any log carbohydrates? So we did sort of a reverse approach for that.
Mm-hmm.
And then we did a similar thing for those, like all of those events. And for each patient, we then sort of tried to identify a pattern, saying like a frame of time where we would normally identify meals. And of course, you would have this like preconceived ideas that breakfast should be in the morning and lunch should be in the middle of the day. But like by random sampling, we had a few individuals that actually worked the night shift, which we actually were available to identify, which was quite interesting. So that's sort of how we went about it. So it's a similar type of training. Like we used the data that was available in order to validate for insulin and carbs. And then we did this manual like interpretations, and then we used that to train a neural network in order to identify those. And then we took sort of so in that sense, the system sort of learned your approach. Like you would most often find meals at these time points. It could be meals at a completely different time point if you were going to a party or you were working the night shift, but most often meals would look like this for that individual.
Thank you.
But of course, I see that was comments on proteins, or if you use different bolusing strategies and so on, but that's not something we use to train the system. But that means that, I mean, you can have, if you would alter your strategy for different meals, your meals can look differently. But that's why we need sort of the like larger amount of data. So you can also have training for the system based on a cohort of patients rather than just your data, in order to cover different aspects of how a meal can look like.
Mm-hmm. Lars, there was a question, and you don't have to share, but why do you prefer MDI to a pump? You saw that go by, didn't you?
I could see that question. Yeah, I would say I think I arrived at quite a good A1C and time and range, even before I started DDA. And now when I'm applying all our methods and algorithms and machine learning on myself, I've actually improved my A1C even further. So I'm currently at 5% with MDI, which I think is quite good. So actually the effort in changing to a pump today, I believe, is quite small, but
Understand. That makes perfect sense. I'm curious. All your clinic and work is done in Sweden. Is that correct?
Right.
Is your diet significantly different in your community than what we in the U.S. would eat, or other places in the world?
I think the answer may be sort of impacted on my view on the US diet. But I would say, at least going back to myself, I keep quite a low carb diet without increasing the fat content. And my general interpretation is actually that the Swedish diet is more low carb than the US in general. But that's excuse me for this stereotype interpretation.
You're not wrong. You're not wrong.
Just as a comment, when we like in the cohort of data that we used in this retrospective trial, like we have the whole range of HbA1c. So we have patients from like the low 5% up to like 12 or 11%. So we've sort of analyzed data from the whole spectra. And of course, we have McDonald's in Sweden as well. So people tend to have very different diets.
And I assume you're an active culture. Most of us are in California, and it's a very active community, but it isn't that way throughout the U.S. As a country, are you an active society?
I think it varies. And Daniel and Per-Ola, you're active clinicians. You should answer about the general picture.
Yeah, well, I would say that, like, in general, in Sweden, people are very active. And I think also for the T1D community in Sweden, we have come quite a long way with the view on diabetes. Of course, there's a lot still to be done, and people still confuse T1D with T2D. And there's this, like, people have preconceived ideas that if you would manage your diet, you would not get type 1 diabetic and so on. So there's still a lot to be done in Sweden, but in general, people with type 1 diabetes are very active, and many are extremely physically active. Some even are elite athletes and so on. And we have a lot of classes on that from the clinic as well, on how to manage insulin when being physically active, and how to even combine T1D with elite athletes and so on. So I would say that it's quite common to have a lot of athletes among type 1 diabetics in Sweden.
In your practices, do you have any loopers?
Yes, but we do not currently have a system in Sweden on how to sort of share that data. So basically, you would have to bring it yourself on your mobile app. So we do not have any clinics that are connected to Nextcalc.
If a patient comes to you and says, I hear about this loop, I want to try it, are you able to start someone on a DIY loop or a closed loop?
Not DIY, but of course, so we can only use those that are sort of commercially available, which would be Tandem or Medtronic for that case.
We're aware of a very, very large group in Norway that has their own Facebook group on looping. I don't know that I've seen anything specifically from Sweden, or whether you have a large enough population that is looping. Not sure about that.
I don't know either. I mean, we know people who are on DIY loop, but I don't know if there's a community specific for Sweden. I don't think so.
Well, tell them about us.
Yeah,
we'll welcome them in. I know we've gone, Daniel, we've gone back and forth about where you are and what your process is. How soon do you think you will be a commercially viable product, and what's your path going forward in terms of what your development will be and what you want to add to your system?
Should I take that? Yes, we're currently started on 2 clinics here in Sweden. So we started with the clinic side of the interpretation. So we do that, and we are expanding. We have a lot of discussions ongoing with some European clinics as well outside Sweden. The plan is to launch the patient mobile app in May, as we were saying, and also to connect patients within Europe still, because we're keen on we need to stay on the CE mark on the regulatory territory where we are, and then to grow the community on using our patient app as well. So gradually moving within the CE marking region. And as we said, we have some ideas and some discussions on how to get the FDA approval as well. But we cannot commit to say that we will do this year.
Good luck with that. We hear they're interesting to work with.
Yeah.
I want to thank you. I think we want to push you to do more at the tighter end, but we totally understand helping those who really do need the help. And I keep seeing the comments going, you can use us in your trials. You can ask us. We'd like to engage with you in ways that are valuable to you, and how we can contribute. Please let us know. And I'll certainly send you all the questions that were in chat so that you have them as maybe guidelines or questions. But we would love to continue to engage with you guys as you move forward.
Thank you. Thank you.
And thank you for taking your time. I know it's late. I think you should go to bed real soon. Thank you so much. Thank you for taking the time, because I know you're real busy. I will have this video up and available, and I'll send you all the information, and we'll be watching to see what you end up doing.
Thank you very much. And it was super interesting to take part of this.
Thank you so much, Lars, Per-Ola, and Daniel. We really appreciate it.
Yeah.
Bye-bye.
Thank you very much.
Have a nice
Thank
day.
you.
Bye. Bye. Bye.
Bye.